Welcome to this module on plasmid DNA. This module will focus on teaching you about the fundamentals of plasmid DNA. What is a plasmid? A plasmid is an extrachromosomal DNA. It replicates independently of the chromosome. It encodes genes which do not have essential functions, and it encodes genes which confer an advantage under specific conditions. The size of a plasmid can range from 2 kilobases to 100 kilobases. A plasmid is negatively supercoiled. The copy number of a plasmid is variable and determined by the type of the plasmid as well as by the environmental conditions. Plasmids have a specific host range. They contain an origin of replication, and their designation is as lowercase, as in PUC19. Plasmids can be supercoiled. They can be relaxed by nicking to give a circular topology, which means that one strand of the plasmid can be nicked using an enzyme such as a nickase to yield a relaxed plasmid. Plasmids can be linearized using restriction enzymes which cleave a specific site within the plasmid DNA. Some of the naturally occurring plasmids are PCOL-E1, which is responsible for the production of bacteriocin, PTOL, which is found in Pseudomonas putida and is responsible for the degradation of aromatic compounds, PTI, which is associated with Agrobacterium tumefaciens, which initiates tumors in plants. In addition to these, plasmids can confer resistance to antibiotics. The copy number of plasmid is determined by the concentration of antibiotics in the culture media or in the environment. Plasmids replicate by two methods. The first is by theta replication, which is bidirectional, and the second one is by rolling circle amplification. Plasmids do not contain proteins for their replication. They do not encode genes associated with replication. Rather, they rely on the host cell machinery to replicate. In the case of rolling circle replication, plasmids rely on the cellular DNA polymerase tree, DNA ligase, and RNA polymerase. Plasmids are classified as relaxed and stringent on the basis of their copy number per cell. Relaxed plasmids are present in a high copy number per cell, and stringent plasmids are present in a low copy number. The copy number of the plasmid is regulated by a feedback mechanism in which RNA molecules such as RNA1 and RNA2 may be associated with regulation, as well as proteins such as ROP1. If the copy number of the plasmid increases, the number of RNA1 transcripts increase, which in turn regulate the RNA copy number by interacting with RNA2. Both RNA1 and RNA2 are plasmid-encoded RNA transcripts. The copy number is regulated by the origin of replication in the case of PUC, PACYC, and PSC101 plasmids. Mutating the DNA sequence encoding the origin of replication will in turn affect the copy number of the plasmid. Plasmids, which are shuttle vectors, can replicate in two hosts. In the case of the plasmid pClac1, the host is a bacteria, E. coli, as well as the yeast Chloromyces lactis. This is because shuttle vectors contain two origins of replication. Thank you for watching this module on the fundamentals of plasmids. Please complete your evaluation at the end of this section.